Hello everyone. In this segment of excretion, we are going to study about mechanism of urine formation. Urine is formed as a result of three main steps. The first step is ultrafiltration. The second step is selective reabsorption. And the third step is tubular secretion. These three steps takes place inside the nephrons, which are present as the structural and functional unit in each kidney. Millions of nephrons are present in each kidney. The nephrons are also known as the uriniferous tubules. So the three steps occur at different parts of the uriniferous tubule. Now we will study each of the step individually. Now this is the first step, which is known as ultrafiltration. This takes place in the Bowman's capsule and the glomerulus. The Bowman's capsule and the glomerulus together is called as the renal corpuscle or Malpighian body. So the first step of mechanism of urine formation, ultrafiltration, takes place in the Malpighian body of the nephron. We have already studied the structure of nephron. Now if this is the Bowman's capsule, the afferent arteriole enters in, bringing the blood, and then this branches up into several capillaries. This forms a network. There are so many capillaries that are present, and all of these capillaries unite to form the efferent arteriole. See the lumen of the arteriole. This is efferent arteriole, and this is afferent arteriole. And the bunch of capillaries is known as glomerulus. So the afferent arteriole comes from the renal artery, and this is the direction of the blood. This comes in, and then since the lumen or the diameter of the blood vessels is gradually decreasing, there is an increase in the pressure, and therefore the liquid part filters from the glomerulus into the Bowman's capsule. This process is known as ultrafiltration. Now there are several pressures that are present over here, which we will be seeing just now, due to which the filtrate is formed. Once the liquid part of the blood gets filtered, it is over here, we call it as the glomerular filtrate. The glomerular filtrate is present over here, and this has been formed from this blood itself. When the blood from the afferent arteriole is coming in, it has the hydrostatic pressure. The hydrostatic pressure of the blood is equal to 60 millimeter of mercury. We always measure the pressure in terms of mercury. So it is the pressure by which the blood is entering through the afferent arteriole. And there, since the blood has many plasma proteins, and inside this glomerulus there is one type of protein which is known as albumin. Now the osmotic pressure, the liquid that is present inside as plasma inside this blood vessel, and this albumin creates an osmotic pressure, and this osmotic pressure has a counter effect. So the pressure which I am talking about is inside the glomerulus. So osmotic pressure in the glomerulus, and this is basically because of the plasma proteins like albumin. I'll mention due to the plasma proteins like albumin. is almost 30 of mercury, Hg. So this is counteracting. The pressure which is over here is 60, but this is opposing to it, and that is why I am using the word that this is a counter pressure. So this is opposing this pressure. This is the first pressure which is counteracting with this pressure. The second pressure that is acting on this is the glomerular filtrate. Now this filtrate which is accumulated over here also creates a reverse effect. It gives a opposite pressure to the filtration process, and this is we call as the hydrostatic pressure. Again, hydrostatic pressure of the filtrate, this glomerular filtrate which is present in the Bowman's capsule. This is almost 10 Hg. And there is one more pressure that is present, which we call as the pressure developed by the interstitial fluid. Now this interstitial fluid is present nearby. The nephrons are present in this interstitial fluid. This fluid also gives a reverse pressure. So this is present outside, and this is also equal to 10 Hg. So the three pressures, the osmotic pressure in the glomerulus, the hydrostatic pressure of the filtrate, and the pressure that is exerted by the interstitial fluid, all of them are against or are in reverse of this hydrostatic pressure of blood. So if we add this up, it comes out to be 50. So the complete hydrostatic pressure is 60 of mercury, and if we minus it, then the pressure of ultrafiltration comes out to be, the net pressure comes out to be 10 of Hg. Now let us see the second step, which is called as the selective reabsorption. Out of the 180 liters of the glomerular filtrate which is produced, hardly 180 liters of the filtrate is produced in 24 hours. Hardly 1.5 liters of urine is formed. Urine is formed daily. So much of it, that means more than 80% of the filtrate is reabsorbed, and that is what we are going to study in this second step, that is the selective reabsorption, in which the filtrate is reabsorbed from of all the useful substances from the nephron. So let us see what all is reabsorbed. There are substances which we can categorize. The first category is high threshold substances. Now, high threshold substances are those substances which are not at all wasted. These are reabsorbed completely. That means 100% of reabsorption takes place, and so we say that they are of high threshold substance. 100% reabsorption occurs of glucose and amino acids. So these are high threshold substances. Then there is low threshold substance, in which urea is an example. Urea, however, is the nitrogenous waste whose removal is done in urine, but actually urea is also reabsorbed in minute quantities, and this maintains the osmotic concentration, and this helps in reabsorption of water also. This urea which is reabsorbed can be recycled again, and thus it is of low threshold substance. And there are certain substances which are zero threshold substance. These are not at all reabsorbed, and these are uric acid, hippuric acid, creatinine, etc. So these are the various types of substances that will be absorbed or will not be reabsorbed in this second step. So let us see in the nephron how this process takes place. So let us see the process of selective reabsorption. Now we know that reabsorption can be of two types. It can be actively done by the expense of ATP, active reabsorption, and passive reabsorption. The passive reabsorption is by diffusion and osmosis. And we know the process that in passive reabsorption, the diffusion is the movement of molecules from higher concentration to lower concentration till both the concentration becomes equal, and therefore the complete reabsorption cannot take place by passive reabsorption, because when both the concentration becomes equal, till that level only the reabsorption will take place. But with the expense of ATP, the active reabsorption takes place. This starts in PCT. The entire step of selective reabsorption, the second step that we are talking about, occurs in PCT and in the loop of Henle. So the filtrate which comes into the PCT has many glucose molecules, amino acids, drugs, and several important molecules. So here the glucose and the amino acids are actively reabsorbed. So this is by active reabsorption. With the expense of ATP, not a single molecule of glucose is wasted. Now again, it has to be seen that it can be only till a particular level, the threshold level of the blood. We know that there is a maximum amount of maximum limit of the blood to hold glucose. Beyond that, the glucose cannot be reabsorbed, and that is why the diabetic patients excrete the glucose in the urine, which otherwise should have not been excreted. So that is a diseased condition in which the glucose is not reabsorbed because that has gone beyond the threshold level of the blood, so it is being excreted. In a normal person, which we are talking about, in a healthy body, the glucose is not at all wasted, so it is totally reabsorbed in the PCT with the expense of ATP, that is the energy molecules. So obviously here the PCT has brush bordered epithelium, and this epithelial cells have high quantity of mitochondria. This mitochondria only generates the ATP, and thus active reabsorption takes place in PCT much because of the high amount of mitochondria in these cells that make up the PCT wall. Then also sodium, about 70 to 75% of sodium and potassium ions are also reabsorbed over here. So the ions are also reabsorbed in PCT. Then comes the loop of Henle. We already have studied that there is a descending limb of loop of Henle and the ascending limb. The thickness of the wall of both the limbs differ. The descending limb is thin and quite permeable to water, while the ascending limb is thick and impermeable to water. So when the filtrate runs down into the loop of Henle, the maximum reabsorption of water takes place. From here it is reabsorbed. So the water is reabsorbed by passive transport. So passive transport, water is reabsorbed because there is abundant water over here. There is no need to expend energy, and water is reabsorbed continuously. And while it is coming down, since the wall is very permeable to water, therefore the filtrate gradually becomes hypertonic because water is getting lost and the filtrate is becoming hypertonic. So if a question comes, this comes usually in NEET etc. or in your exam also, ki how is the nature of the fluid or the filtrate that runs down the descending limb? It's hypertonic. And why is it hypertonic? Because the walls are so permeable to water that the water is reabsorbed by the kidney cells, and so then it comes over here. So here, till here, the limb is quite permeable, but then after that, when it moves into the ascending limb, the filtrate does not lose water at all. In fact, it becomes isotonic because the walls of the ascending limb become impermeable to water. So here only the ionic absorption takes place. Whatever leftover sodium ions and potassium ions, here 70 to 75% was reabsorbed in PCT, and rest of the sodium ions, potassium ions and bicarbonate ions which are necessary for the body are reabsorbed from the filtrate while it is moving up. There is no reabsorption of water in the ascending limb of loop of Henle. So in both the limbs of loop of Henle, it differs. Here there is no water reabsorption, and here it is only the reabsorption of water. So this is what is the selective reabsorption. I have drawn this because this particular portion, the PCT, DCT and the renal corpus lies in the renal cortex. If you remember the longitudinal section of kidney in my previous lectures, I have explained that there are two major portions, the renal cortex and the renal medulla. So the renal medulla is having this loop of Henle as well as the collecting duct. So according to the length of loop of Henle and the position of this particular part, if it is very small, the loop of Henle is very small and it almost ends up in the cortical region, then such nephrons are called as cortical nephrons. In such organisms, much water reabsorption is not required. So this is usually in aquatic organisms because the loop of Henle is meant for the reabsorption of water only. But in terrestrial organisms like us and all the other animals which are living in desertified areas, they have juxtamedullary nephrons. These nephrons have very long loop of Henle. That means the renal medulla has very long loop of Henle. The longer the limb, the more is the reabsorption of water, because their body do not want to waste the water at all, and therefore they completely reabsorb the water. So such type of nephrons in which the loop of Henle is quite big and is innervated much into the renal medulla part, those are called as juxtamedullary nephrons. In human beings, we have 3/4 cortical nephrons and 1/4 juxtamedullary nephrons. So we have a combination of both the types of nephrons. So this is what is the selective reabsorption. Now let us see the last step of the mechanism of urine formation, which is the tubular secretion. Now this is the efferent arteriole which comes out of the Bowman's capsule. This further branches to form many capillaries. So there are many capillaries over here, and these are called as peritubular capillaries. This particular step, the tubular secretion, is so called because this occurs in the tubules. Now here we have two tubules, the DCT and the PCT. In these two tubules, the waste material or the unwanted substances are removed from the peritubular capillaries. There are still much more nitrogenous waste and other substances that are present in the blood even after filtration. So after the filtration, the blood is moving in this from the efferent arteriole, which further forms the capillaries called as peritubular capillaries. From these peritubular capillaries, the waste substances are secreted into the PCT. Now as I told you that this has the brush bordered epithelium, and there is also certain glands that are present in this and in this, and therefore suppose this is the capillary which is pouring all the waste into the DCT. So the waste materials from here are secreted into the tubules, and then these cells of the tubules add the waste to the filtrate. So the secretion of these wastes is added up to the filtrate, and only when the secretion takes place in the DCT, then the filtrate is rendered as urine. Before that, we call the filtrate as the nephric filtrate because it is traveling in the nephron, which is having good amount of waste, but we call it as urine only after the secretion from the DCT takes place. So the tubules are secreting all the waste substances from the peritubular capillary capillaries into the filtrate. This is known as tubular secretion. So what are all the ways? As earlier also I discussed, these are still more ways like hippuric acid, creatinine, excess amount of sodium ions, and many more ions which are not required, especially the hydrogen ions, ammonia, ammonium ions, etc., which are not required, are secreted, and then the fluid is rendered as the urine. So this takes place in the DCT, and then from the collecting tubule it runs down the collecting duct to open into the renal pelvis and out from the ureter into the urinary bladder. So this is what is the third step that we call as tubular secretion. So this is all about the mechanism of urine formation. In my next video I'll be dealing with how the urine is regulated, the regulation of urine formation, and certain other steps like, and certain other things like artificial kidney or dialysis and counter current mechanism. If you have any doubts in this video, you may ask me in the comment section, and you can also refer to the questions given in the description box. Thank you.